So earlier we spoke about complete dominance, where either allele must be dominant completely over the other allele. So that's where you have only one phenotype of the two being expressed. So if you had a red flowering plant crossed with a white flowering plant, the offspring can only ever be red or white, right? That's complete dominance. Now we talk about incomplete dominance, where both alleles are incomplete dominance, dominant, and they both express themselves in the phenotype. For example, a gray rabbit, yeah, being the offspring of a black and a white rabbit. So let's do an example of that, shall we? Start setting it up. Yeah, right, incomplete dominance. So both are homozygous, of course. Then our genotypes should reflect that. So the black will be BB. Now the white is not a recessive trait of black. The white is not an allele of black. Okay, a white is just as dominant, so it both gets a W, capital W, because it is dominant. Okay, so you name with the dominant trait, and then we have meiosis taking place. Okay, remember, nice meiosis. Okay, and so we have gametes forming that can either carry the dominant black traits or the dominant white trait. Okay, so these become your gametes. Don't forget to label them gametes in this line. After gametes we have fertilization taking place, right? Fertilization, so you can use your Punnett square, or you can do this, and you can say we have black and white, BW, black and white, we have black and white, we have black and white. Okay, no, sorry, this to come from here. Yeah, black and white. You can also use a Punnett square, that's a more reliable method, but this works as well. Make sure you know where your arrows are coming from. Then you write down phenotype. This will now be your F1, your first filial generation. Got it? So this is, yeah. And then you can write down black, white, or black, white. And your genotype, sorry, it's genotype first, and then I show you genes. And then you explain the phenotype based on the genotype. So you always extrapolate your phenotype from the gene sequence that you listed above. Okay, so phenotype, and here the phenotype is going to be gray. So your offspring will be gray in color, right? Same thing if you did it with the plant, because BW means gray. If you cross two gray rabbits, let's see what happens. Okay, two gray rabbits are crossed. So we have P1, or rather P2 from the F1 generation, okay? P2, phenotype, gray crossed with gray. Genotype of that would be black, white crossed with black, white, right? Meiosis occurs, meiosis, okay? And we have gamete formation. One black, one white, one black allele, one white allele. Right? That's just how it is. Then we have fertilization taking place. Right, fertilization occurs. And let's fertilize. So black sperm, black egg, BB. White sperm, black egg, BW. Black sperm, white egg, BW. And black sperm, white egg. No, that's correct, yeah. BW, one coming from each. Yeah. So, no, that's wrong. It should be, yeah, it should be that. WW, yes. So your F2 now, right, where we have your genotypes, would be one black, right, is to 2 BW is to 1 WW, white, white. And then our phenotype expressed is therefore going to be a black rabbit, a black offspring, is to 2 grey offspring, is to 1 white offspring. So the chances of having a white child from the grey rabbit and from 2 grey rabbits is 25%. The chance of having another grey rabbit is 50%. Right, hope you understand how the probabilities work. Right, so genotype comes from the Greek word meaning genos, or to descend or race. Okay, and constitutes, it's the genetic constituent of the organism, genotype. Okay, the chemical, genetic constituents of the organism. Okay, whereas phenotype, coming from the word phainin, right? Phainin, rather, phainin, but phenotype, right? That phainin means to appear or show. Phenotype. So not outward appearance, no. You will say observable, physical characteristic. Remember that characteristic could be resistance to climate, resistance to bacteria. It could be, um, um, you know, tolerance in terms of nutrient deficiency and stuff like that. Right, so alleles, also known as an allelomorph, is already explained as that single characteristic for a gene coding for a characteristic. Okay, we'll talk more about dihybrid crossing. Let's do a dihybrid cross then, rather. Okay, it's a little bit more of an issue, but we use a Punnett square, a very large Punnett square for dihybrid crossing. Let's do that though. A dihybrid cross, when we're looking at two pairs of contrasting characteristics, so two pairs of contrasting characteristics. Monohybrid was a single pair of contrasting characteristics. Dihybrid is two pairs of contrasting characteristics. Let's look at what that means. We have a so what are our two characteristics in this case? Let's look at a plant. Let's call it red. So we're going to look at plant color. Our two characteristics in this case are going to be plant flower color, either red or white. Okay, red dominant, white recessive. Okay, and we're going to look at plant height. Plant height. We're going to look at tall and short. Okay, so just to show this, we're going to speak about redness, so big R, and small r. Okay, just to show this, we're going to speak about big T and small t. So red flowers, tall, short, that kind of combination. We're going to look at it together. Okay, so let's look at how we go about doing this. First, so let's understand that in our cell, okay, in our cell, we're going to have an allele or a gene component for big R, an allele or gene component for small r, an allele or gene component for big T, and allele or gene component for small t. Okay, sorry, that should be small t. Then when meiosis occurs to this big cell, right? So what happens? Okay, so we can have R and then T, right? And then here we have R, and here we have small t, pulled to the sides, right? Or we can have this happening. It's a very large cell, let's make it roughly the same size. Okay, we can have big R and big T, and small r, small t. as our combinations, right? Which means the cell that form from here is going to be 2. This cell will have big R and big T, or it can have, sorry, big R and small t. Okay, big R and small t, or it can have small r and big T. Okay, and from here we can either look at the combinations, please. We can have big R and big T, so it can be red flowers and it can be tall, or it could be white flowers, right, and it can be short. Okay, so that's just how the combinations work diagrammatically if you had to look at it through meiosis. Okay, because that's what's happening here. We're having random segregation, random assortment. Please read up on Mendel's two laws and learn that by heart. Mendel's laws just explained what I explained in the previous video about the meiosis occurring here. That's all. Just understand it conceptually. They can ask you for definitions, so learn that by rote. But conceptually, you already understand this. That from here, these genes, we get one allele and a second allele from meiosis. That's all it's telling you. And the random assortment is just this portion here. It's all just crossing over. So that's all that's happening. Not crossing over, but being fertilized, and the crossing is happening. Right, so let's do this as you would a Punnett square form when you asked to do a calculation of sorts, okay? So, yeah, it's the same thing that I just showed you in the diagram, but just more formally written out, and it can look very confusing, very confusing, okay? So, yeah, let's go for it, shall we? Mendel's second law is the law of independent assortment, by the way. So stated, each pair of contrasted characteristics may be combined with either of another pair. Simple. That's exactly what I showed you in this part here. The black white thing, where each gets a black or a white, that kind of nonsense, right? Straightforward. There's nothing hectic going on there. So I should probably show you now. So let's say we have one plant. So our first parental generation, we're going to start with a phenotype of our organisms, and that phenotype is going to be a tall red flowering plant crossed with a short white flowering plant. Okay, tall red flowering plant with a short white flowering plant. Now we don't need all the genetic information, because if we were told what's recessive and what's dominant, then we're sorted, and we have been. So genotype is going to be for tall and red. It's going to be 2 dominant alleles for tall. Oh, wait. So we can have T and we can have a small t. So we need to know whether this tallness is homozygous or heterozygous for tall. So let's say homozygous. In the question we were given homozygous, tall and red. Okay, homozygous, tall and red. We're going to have TT, RR. Okay, remember we're looking at 2 pairs of contrasting characteristics, tallness and color, crossed with shortness, which is going to be TT, and don't put a thing there, just leave it, right? And it's going to be RR. These are the genotypes that we have. Meiosis is going to take place. Meiosis, okay. Meiosis, lovely meiosis. And then we have the gametes that are forming. So we're going to have a gamete with big T, a gamete with another big T, a gamete with a big R, another gamete with a big R, and we're good to go, right? Then we have from here a gamete with a small t, another gamete with a small t, small r. You can sort of see where we're going. It's not hard, just get used to practicing it, okay? And then we have fertilization going on there. So this is fertilization. Yay. Now it'll be pretty hectic for you to do this. Yeah, it's going to end up a lot. You're going to have 16 of these, and it's just going to take... Nah, so what we use is a Punnett square, okay? So on this side, we have T and R, okay? So one from each, T and R, okay? Now before we do that, actually, Yeah, now we need to have our combination set up. So we can have a big T with a big R, we can have a big T with a small r, or we can have a, yeah, big T, we can have a small t and a big R, or we could have a small t and a small r. These are the combinations of gametes that we can expect. Okay, then we use that in our Punnett square. Now this is derived just simply from there. We can have a big T with a small big R. We can have a big T with a small r from the side. Okay, and that's really about it. Okay, it's pretty not hectic there. Yeah, that's fine. Because what we did, let me just show you that again. Might not have been clear. So the only possible combinations of tallness and shortness that can, oh sorry, of height and color that can happen here is we can have a big T and we can have an R. Then here we can have a small t and we can have a small r. That's all. And this is actually what we base our crosses on. So we say t. So we have t, but we don't do that. We combine the t and the r, right? So I hope you understand that. So we can say t and r, t and small r, right? And we can say small r and t, big R and T, and this becomes 4. 1, 2, 3, 4 combinations. Okay, and then yeah, so let's write that all down, and we get this. Yeah, T R T R T R T R, right? And this is what goes in our sides on our Punnett square. This is the only part that tends to confuse students, but once you see it in action, then you'll be fine. All right, trust me. So we get TR. Okay, bear with me. Okay, trust me, this is a very nice section. You will find it pleasant. Don't fret about that. So we have now TR there, we have TR there, we have TR there, and we have TR like so, right? And we can just do that. And it looks a bit different from the other one, isn't it? Okay, now we can start to do our thing. Same thing. Let's combine all of these from going left to right. So we have TR, 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 TR. Okay. If it makes you feel comfortable, we do them one at a time. So here you can take your time, do it slowly. Yes, they will ask you to do this in the exams, right? They will ask you to do it. Take your time, take your time. Let's do all the T's, then we do all the R's. Okay, so here we have a big T from the top, and we have a big T from the side. Okay, here we have a big T from the top, big T from the side. Here we have a big T from the top, small t from the side, big T from the top, small t from the side. I hope you can see that. That's where I got my information from. Okay, like that. I've just been doing that for each thing. Okay. Okay, cool. Now let's do the T's on the side. Big T from the top, big T from the side. Big T from the top, big T from the side. Big T from the top, small t from the side. Big T from the top, small t from the side. Small t from the top, big T from the side. We know our order, it's capital first. So big T from the top, small t from the side. Sorry, small t from the top, big T from the side. That's correct. Small t from the side, big T from the top. Small t from the top, small t from the side. Small t from the top, small t from the side. Small t from the top, big T from the side. So order, remember, it helps to do this in pencil first and then do it properly, right? So big T, small t. Here we have another big T and a small t. Here we have a small t and a small t. Here we have a small t and a small t. Now we do our R's, okay? So it's going to be R, R. You can do this, like, pretty alright. R, 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 okay? Here we're going to have R, 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 small r, and here we're gonna have just red and then white. Okay, so here we're gonna have red and red, red and white. Here we're gonna have red and white, and here we're gonna have red and red. Here we're going to have red and red, sorry, red and white. We're gonna have white and white, white and white, red and white. Got it. So these are our combinations. Now we look at defining these, okay, or like calling them out. So this is our first filial generation. Let's write down our genotypes. Okay, so all our different genotypes. Let's look for okay, so tallness and red. Let's look for what's tall and red. Okay, so tall is going to be tall and red, which means big T and big R. This is going to be tall and red. This is going to be tall and red. This is going to be tall and red, and this is going to be tall and red. This is also going to be tall and red. This is going to be tall but white, so we leave that out. This is going to be tall and red. This is going to be tall but white, so we leave it out. This is going to be tall but red, tall and white, tall and sorry, short and red, so not going to help us. Short, not going to help us. Tall and red, short and red, short and red. So how many ticks do we have? 1, 2, 3, 4, 5, 6, 7, 8, 9. So we can write there 9 tall red, right? Now let's look for a different thing. Let's look for tall and white. So this is look for big T but small r's there. So like here, tall and white. Okay, tall and white, tall and white, and mathematically that should be about it. So we have 3 tall and white. Okay, then let's look at short and red. Got it. I know it can get a bit hectic, isn't it? Short, so we can leave out what we've ticked already, isn't it? So we can ignore these first two columns already, and we're looking for short. So small t's. Here's small t and red. Okay, that's right. Okay, so that's in the middle. So that's going to be short and red. It's going to be short and white, not what we want. Short and red, correct. Short and red, nice, correct. So it's going to be 3 short and red, okay, is 2, and then we our last thing there. Let's do it in like lights. No, we've done that. Brownish thing, right? The only thing that's left is short and white, and there's only one of those happening, right? So we can have 1 short white. That stands to reason because shortness and whiteness are both recessive, so they should be in the smallest proportion, and tallness and redness are both dominant, so they should be in the largest proportion. So this makes sense. So the order is 9 3 3 1. Okay, you'll see in multiple choice that you might need to know that as theory, but you'll see it through calculations. Okay, so that's how you do a dihybrid cross. All right.